Okay, so we just learned a lot about water and the water properties and why water is important to living things. And now we're going to move into chapter 4, which is about carbon, the element carbon. And the reason why is because most of the other compounds that are really important to living things, carbon is the main element or the backbone of that molecule. And so therefore, we're going to learn about some properties of carbon and some different chemical groups, so that when we get into chapter 5 and look at the different types of molecules that there are, that we can know some things about the molecule based upon these chemical groups that are attached to it. So the overview about carbon. Carbon is the backbone of most biological molecules. And so when we look at DNA and carbohydrates and proteins and lipids and so on, it's a big element. So although cells are 70 to 95% water, we learned that last chapter, the rest consists mostly of carbon-based compounds, as I just said. So carbon is unparalleled in its ability to form large, complex, and diverse molecules, and we'll look at why that is. So all of our major macromolecules, macro means big, proteins, DNA, carbohydrates, other molecules, are all composed of carbon, what we call skeleton or carbon compounds. So the study of chemistry that deals with carbon-based molecules is called organic chemistry. So organic compounds range from simple molecules to colossal, very big ones. And so we go from small to big. Organic means that it contains carbon, and so therefore it's different than in layman's terms when people talk about organic food that was grown without fertilizers or pesticides and sprays and things like that. This is unique in chemistry and therefore in biology as well. Most organic compounds contain hydrogen atoms in addition to carbon atoms. And so this is an old term, vitalism. The idea that organic compounds arise only in organisms was disproved when chemists synthesized or made the compounds in the laboratory. And so therefore carbon compounds can be made within cells but also outside of cells as well. And so mechanism is the view that all natural phenomena are governed by physical and chemical laws, and we'll get into that here as we look at carbon. So for carbon atoms can form diverse molecules by bonding to 4 other atoms. So carbon makes 4 bonds. So when we look at carbon, like this here, so in its outer shell it has 4 electrons. It needs 4 more electrons, so therefore can share and make a bond with 4 additional atoms. So it always makes 4 bonds all the time. So electron configuration is the key to an atom's characteristics. Carbon is different than hydrogen. Hydrogen only can make 1 bond because it has it needs 2 electrons for the outer shell. It has 1, so it shares and will make another bond. So it makes 1 bond. Electron configuration determines the kinds and number of bonds an atom will form with other atoms. All right, and so how many valence electrons it has will determine those bond formations. So the formation of bonds with carbon, it has 4 valence electrons. Carbon can form, therefore, 4 covalent bonds with a variety of atoms. And so covalent, remember, means sharing, so it's sharing the electrons, and it has 4 of its own, and therefore needs 4 more, so therefore will share an electron with 4 other atoms, and those other atoms will share an electron with it. And so, like, for instance, carbon can bind to 4 different hydrogen, like this. All right, and so what's happening, these lines represent this carbon is sharing an atom with hydrogen, this carbon is sharing an electron with this hydrogen, and this hydrogen only has 1 electron, is sharing it. So each one of these these lines represent a shared pair of electrons. So carbon has the 4 original electrons, and it gains 4 more by bonding to the 4 additional hydrogens. So we call this tetravalence. Tetra means 4. So this tetravalence makes large complex molecules possible. So 4 valence electrons, and it needs 4. It makes 4 bonds. So in molecules with multiple carbons, each carbon bonded to 4 other atoms has a tetrahedral shape. And so what that means is if we turn the page here, it has, this is a tetrahedral shape. So here's the 4 bonds. This is CH4, so C in the middle, and then the 4 bonds. And so realistically, the bond angles are like this. All right, and those are your 4 bonds. So here's your carbon and your 4 hydrogens. You should remember that from chemistry, the shape. All right, and so, however, when 2, let's go back up here so we don't forget that, when 2 carbon atoms are joined by a double bond, the molecule has a flat shape. And so if we look here, this represents a double bond down here, this picture. And so this is C2H4. So when you have 2 carbons and only 4 hydrogens, so you have each carbon bonded to 2 hydrogens, this can't be a single bond because remember each carbon needs to bond to make 4 bonds. And so therefore only 2 go to the hydrogen. That means they double bond to each other. And so that's what this double bond here is showing you here, this double bond, and these are the hydrogens here, and therefore have a flat shape. And so it's in like kind of one plane there. And so therefore the way that they bond and the elements that are formed and the way that they bond determine shape. And we'll see as we learn more about different molecules that shape helps to determine function in living things. And so the shape of a molecule is very, very important. The electron configuration of carbon gives it covalent compatibility with many different elements. And so it can make 4 different bonds and go to 4 different elements or all the same, like in this particular case here. The valences of carbon and its most frequent partners. The most frequent partners are hydrogen, oxygen, and nitrogen. And so those you see the most, most living things, it's like this Chan, all right, carbon, hydrogen, oxygen, nitrogen. These are most frequently, but we can add a PS here as well. And so there are phosphorus and sulfur as well, and these are your 5 most common elements in living things. And so this is its most frequent partners there. And so this is showing you those 4 frequent partners here. Here's your carbon. This is showing you that this is the nucleus. It would have the neutrons and protons, and these are your electron shells. And so carbon has 4 electrons. So therefore, to have this shell filled, it needs 1 more here, 1 more here, 1 more here, and 1 more here. So it needs 4 more electrons, so that's why it can make 4 bonds, so that 4 different elements can share their electron with it, and so therefore carbon always makes the 4 bonds. Here's nitrogen. Nitrogen has 5 electrons in its outer shell, can fit 8, so it needs 3. So nitrogen would make how many bonds? 3. And then oxygen has 2, 4, 6, and it can fit 8, and therefore can make 2 bonds. And so hence H2O, think of H2O here, 2 bonds. And then hydrogen fits 2 in that first shell here, and so therefore only makes 1 bond. And so the molecular diversity arising from carbon skeleton variation. So carbon can bind to all these different types of elements, and the carbon chains, when the carbon is hooked to another carbon, is called a skeleton. Forms the skeletons of most organic molecules. These carbon chains vary in both length and shape. So if we look at the next page, this shows you this. Let's look at a particular color here. The shaded parts here that we're looking at, different molecules here. The shaded part is the carbon skeleton. So this, let's write here, equals carbon skeleton. So we look at how the carbon skeleton differs. So in this particular case, the carbon skeleton, these two differ in length. So this is showing you, we wrote here that the carbon chains vary in length and shape. So we're seeing here the different lengths, 2 versus 3. And so here are your carbon skeletons for these guys. Here, and notice here. And so this is the shape. This is what we call, so this is linear. We call this branching. And so, this is, let's write here that these guys are branching. Linear versus branching. So sometimes you can get that. And the next one, look at here, the carbon skeletons are in, here's a ring. Okay, so the carbon skeleton. So let's write this in rings. So if we look at here, these two, different length, these guys are branching. Let's look at these two here. What I'd like you to do is write the molecular formula of these guys underneath it. So so C something, H something, that's their molecular formula. So pause this and do that for me. So you should have, when you pause this, let me get a different color here. So we have C, 1, 2, 3, 4, and H, 4, 8, 9, 10. C4, H10. If we look here, these guys have 3, 4, so C4, 3 hydrogens here, 3 here, 6, 7, 8, 9, 10. So notice here, H10. And so these guys are actually two different molecules, but they have the same formula. But yet what's different about them is their structure. And we call these guys isomers of one another. That's what an isomer is. And so this is actually called butane, and this is called isobutane. Iso referring to isomer. And so therefore, you can have the same molecular formula but be put together differently because of the way that the atoms within the molecule bond to one another. So all of these in the various pictures here, all are just made out of two elements, carbon and hydrogen. And so therefore, these two elements, molecules that are just made out of those two elements, have a special name. They are called hydrocarbons. And so hydrocarbon, the word hydrocarbon comes from hydrogen and carbon, meaning that the molecules are just made out of carbon and hydrogen. So many organic molecules, such as fats, have hydrocarbon components. And the hydrocarbon components, the bond between hydrogen and carbon, we're going to see later on, has a lot of energy. And so therefore fats have a lot of energy. We have a lot of energy within our lipids. Hydrocarbons can undergo reactions that release a large amount of energy. And so this right here is a picture of a type of lipid. And so this right here is let's do it like this here. This is a fat molecule. And each one of these, so you can think, well, it has a backbone like this, and it has these little chains coming off. These dark ones are the carbons, and these are the hydrogens coming off of it. So all three of these are what are hydrocarbon chains. And these guys, as we said, can undergo reactions where we can break them down. It releases large amounts of energy, which is why fat has so much energy, which is why we need very little fat in our diet, because there's so much energy within a fat molecule because of these hydrocarbon tails. This right here is a picture. I'm going to show you the actual picture here so you can see the color. And so this is tissue called adipose tissue. Adipose tissue are made out of specialized cells called adipose cells, and their main job is to store fat. And so what happens is when we eat fat, it we digest it, it goes into our bloodstream, it goes into our cells, and goes specifically, the excess fat can be stored in our adipose cells, and it fills up, it has a special vacuole inside that fills up with fat droplets. And the fat droplets, if you look at here, this is one cell, and you can see almost the whole cell is red. They stained the fat droplets red, so the red, the fat just basically takes up the whole cell. So they have this whole big vacuole inside filled with fat. And so any excess fat we consume gets stored in these adipose cells. And so we don't need a lot of this fat in our diet, because, like I said, we get a lot of energy from very little fat. Okay, so we don't need very much. And unfortunately, in our American diet, we get more fat than we actually need for energy to live. Okay, so a molecule, I should say, can be itself a hydrocarbon, so the whole molecule made out of just carbon and hydrogen, or you can have parts of a molecule be hydrocarbon, and then this part is actually not, but we'll learn that more about that in chapter 5. So let's turn the page and talk more about this term called isomer. All right, so I used that example in the previous picture. So isomers are compounds with the same molecular formula but different structures and properties. And so that's the definition of it. So these guys are isomers. So these two, that's right here, are isomers. Because they have the same molecular formula, different structure. All right, so that's what we need to know about isomers. And so this different structure changes the properties of the molecule, which then changes its function and how it reacts with different things and so on. And so that's important in the cell, in chemical reactions that are important to keep things alive. Now, these three right here are examples of different types of isomers. Now I want to point out examples of types of isomers. So there's structural, geometric, and what are called enantiomers. Now for this class, you do not need to memorize this, and I'm not going to give you a picture of two molecules and ask them if they are geometric and enantiomers, but you need to understand that they are all examples of isomers. I will give you some practice with that just so that you can get the idea of isomers, but for a test and so on, I won't say, oh, are these guys geometric isomers? So let's talk about, I have pictures of each one of them. Structural isomers have different covalent arrangements of their atoms. So that is the way that they bond together. So that first picture back over here, these guys would be structural isomers. They have the same formula, and it's just the branching is different, and so the covalent bonds are just a little different and giving it a different shape. So that's what it means by covalent arrangement. And so this example right here, example A, would be like that. So here we have, I'm doing the carbon skeleton so we can look to see how that branches. And so these guys would be examples of structural isomers. And so again, we can count up the atoms. And so we have 1, 2, 3, 4, 5. So both of these have C5 and then H5, 10, H12. And so therefore, same formula, different covalent arrangement of the atoms. And so that's structural. Geometric isomers have the same covalent arrangements, but they differ in their spatial arrangements around, the important thing here is around a double bond. So you're going to see a double bond here. And so basically, this is geometric isomers. These guys here, there's not a element X, and so I want to point that out here. Notice this says X, and this is X here, and then this and this. All right, so the X refers to an atom or group of atoms, and it's the same. So for instance, it could be, X could be to this carbon, that bond right here, it could be an O bonded with an H, so OH, and so the X represents an OH. So when we look at this, we could say, okay, C, and then we have an OH here attached, and an OH here, and an OH here, and an OH here. So it just means that it can be an atom or group of atoms. And so then what's different about this is that geometric has, the carbon has this double bond. The double bond is important because double bonds don't allow twisting. And so what happens is that if these OHs are on the top here, then there's no way that I can rearrange. If I imagine I can physically pick this up, there's no way that I can rearrange this in space to make it exactly like this one over here, where the OHs are on the opposite sides of that double bond, all right, kind of kitty corner from one another. And so if I took this and picked it up and even flipped it over, the H's would be on the top and the X's would be on the bottom. So that X would now be down here, but this X would still be down here, and therefore I can't make it look like that. And so therefore these guys are another type of isomer. They have the same molecular formula, same number of carbons, hydrogens, and oxygens in this case, because I used OH as an example, but there's no way that you, short of breaking the bonds and rearranging the atoms, there's no way that you can make them into one another. Same thing with these guys. Okay, so those are geometric isomers. So you're always looking for that double bond between two carbon atoms. And lastly, enantiomers are another type of isomer. These guys are mirror images of one another. So let's just think about images. Let's just think about when you look in the mirror, what changes. The top and the bottom doesn't. Your head is still on top and your feet are still at the bottom. But what changes is what? Your left and your right, right? And so this is what enantiomers look like. So let's label C is enantiomers. So they're trying to show you that mirror image with your hand. All right, so in this case here, with enantiomers, you have a carbon in the middle. This is called an asymmetric carbon atom. So this right here, this right here, all right, this is your carbon there. This is called an asymmetric carbon atom. All right, what does that mean? It bonds to 4 different kinds of atoms or groups of atoms. All right, so what does that mean? Let's look at the 4 bonds that carbon is making. So in this case here, remember carbon always makes the 4 bonds, so these are your 4 bonds. And notice that the group of atoms are so we look at that bond, and we don't look at the actual atom it's bonded to, we looked at the whole group that's bonded to that. And so we have 1, 2, 3, 4 different groups, and hence this is the asymmetric carbon atom. And then this side over here, this one is also making the 4 bonds, and we have the same 4 different groups. Now notice here when we look at them, the top and the bottom are the same. So you have the carbon, you have the CH3, you have the CO2H here, but what has changed is the left and the right. And so therefore, they've switched sides, and so therefore hence are mirror images of one another, and therefore are enantiomers. And so if we counted up all the atoms, the number of each one of them will be the same, so they are indeed isomers of one another. And there's no way short of breaking the bonds and reforming them that we can arrange this one in space to make it look like this one. A lot of people go, oh, well, let's just twist it. If we imagine we twist it, and so the NH2 is over here, but if we twist it, the NH2 is going to be over here, the H is going to be here, and the CH3 is going to be here, and so therefore it won't look the same as that. And so therefore these guys are enantiomers. All 3 different types of isomers, like I said, if I gave you pictures of this, you would need to be able to tell me that they're isomers of one another. I will not ask you if they're enantiomers, okay, on a test. So you need to be able to just look at whether or not they're isomers. And enantiomers are important in the pharmaceutical industry. 2 enantiomers of a drug. Remember, shape determines function. So 2 enantiomers of a drug may have different, let's turn it over here, have different effects. And so differing effects of enantiomers demonstrate that organisms are sensitive to even very subtle, let's put in here subtle, variation in the molecule. And so if we look at an example here, these are 2 drugs. L-DOPA is an effective medicine against Parkinson's disease, which is a disorder of your central nervous system. The central nervous system. And D-DOPA is biologically inactive, so that means it has no effect. So the fact that these guys are isomers, they have the same molecular formula, different structure, changes how it reacts in our body. So no effect versus a good effect here, and therefore we want to make sure to give the L-DOPA as for people who have Parkinson's. I'm going to give you another example over here. So let's write another example. And this is with a drug called thalidomide. And this drug was given to pregnant women in the 50s and 60s. And so let's write a few things about it, a few bullets here. It actually was a mixture of 2 enantiomers, which is, remember, a type of isomer. So therefore they have same molecular formula, different structures. In this case here, they were mirror image of each other. One decreased morning sickness. And that was the desired effect of giving people the drug, to help them reduce that morning sickness and feeling of nausea in the morning, and can therefore, if they had a job, or they could do what they needed to do. But the other enantiomer that was also mixed in here, one actually they figured out eventually caused birth defects. So clearly not a good effect. All right, and so this is another example of an isomer, two isomers of one another, and one having one function and another one having a completely different, even though they have the same formula yet different shape. All right, so those are some real world examples of that. So then when we're looking at now carbon compounds in living things, there are different groups that can be attached to the molecule. So not all carbon compounds in living things are hydrocarbons. They can have these what are called functional groups. They're part of the molecules involved in chemical reactions and help them to increase reactions and also make them polar as well. So distinctive properties of organic molecules depend not only on the carbon skeleton but also on the molecular components attached to it. So we just talked about the carbon skeleton with hydrocarbons, and now we're going to look at other groups. Certain groups of atoms are often attached to these skeletons of organic molecules. And so if we know these different groups, we can look at unknown molecules and tell some things about them. So functional groups are the components of organic molecules that are most commonly involved in chemical reactions. So the number and arrangement of functional groups give each molecule its unique properties. And so what types of functional groups are attached to them? Some are acidic and therefore make the molecule acidic, and so on. So let's look at here the next page. What we are looking at here, when you're looking at these molecules, these are the bonds between carbon atoms. So when you're looking at a ring like this, the book is going to assume that you're going to know that each one of these points is a carbon atom. All right, so each one of those is a carbon atom. And so they're showing you the carbon skeleton here. And so this molecule is 4 rings that are fused together, meaning that one side of one ring is one side of another ring. That's the fusion part. So we have 1, 2, 3, 4 rings here. And so then what we're looking at here is these different chemical groups attached to it. So this one, and this one. And what we're looking at to compare these two molecules is the location of these chemical groups. And so notice here there's not an OH here, it's a double bonded O, and then there's 2 CH3s, and then this OH is the same in the same spot. This has the same 4 fused rings here. And so the location, these guys, the functional groups, can change the molecule and therefore its function. One other difference you might have noticed is the double bonds in the ring. So they become single bonds and so on. So these guys here, we have these two things. These guys differ in functional groups. That's these OHs and so on. Functional groups attached to the 4 fused rings. And this molecule up here is called estradiol. And this molecule down here is testosterone. And so these are a comparison of female and male sex hormones and how they have similarities in their structure but yet differences, and therefore have different ways that they interact and different things that they cause to happen within the body and then within cells. Estradiol is a type of estrogen. All right, so you've probably heard of estrogen. So let's talk about the different types of functional groups that there can be. So here there are 6 functional groups that are most important in living things. And so these are our 6 here. And so we're going to go through each one of these. And so in this part of the notes, I just copied it from your book, and so there's actually not much to write on, and so I'm just going to talk about it here. So the hydroxyl group is made out of hydrogen and oxygen, so it's OH, so it looks like this. It's really like the O, if this is your molecule, and the O is attached to an H, so that they just don't draw the bond between the OH. And so, so this is what that looks like here. So it would be like this OH like that, is another way to draw that. It's not, oh, it looks like it's attached to this hydrogen. It's supposed to be like this. So this bond right here could be also written like this. All right. These guys can interchange and are the same thing. All right, so this is the name of any molecule that has the OH on here is considered an alcohol. So this is ethanol, found in alcoholic beverages. And this gives you some of the functional properties. So it's polar. So when you have an OH on there, it increases the polarity of the molecule. Hydrocarbons are non-polar molecules. So hydrocarbon, if this is, so this is this, compared to, all I did is change here, the OH and the OH here. So this is a hydrocarbon, and hydrocarbons are always non-polar. Hydrocarbon, this is non-polar. That's because the bond between carbon and hydrogen is a nonpolar bond, and so therefore there's equal sharing of electrons and no charges on the molecule. And so here, this is not a hydrocarbon. We added the OH, so this increases the polarity of this molecule and increases its water solubility. So it attracts water molecules, helping to dissolve organic compounds like sugars and so on. All right, and so that's the hydroxyl functional group. Carbonyl functional group has a carbon with the double bond to an oxygen. So that's what it looks like physically. So there's two different names. The names of the compounds could be either a ketone or an aldehyde. And so when we look at this, it depends on where the carbonyl group is. So we have two different examples here. And so notice that this says this is a ketone here. So that's example 1. It says if the carbonyl group is within a carbon skeleton. So notice, where's the carbon skeleton? It's right here, right? This is the carbon skeleton. Notice that it's in the middle carbon atom, the carbonyl group. So right here, that's your carbonyl group. And so it's in the middle, and so in middle, or you can think of it as or not on the end. And so therefore that is a ketone. So the group attached to it is a carbonyl group. The whole molecule is called a ketone. And so this is an actual picture of acetone. And then here we have an aldehyde. So now if we look at, here's the carbon skeleton. Notice here's your carbonyl group, and the carbonyl group is at the end of the carbon skeleton. So it's at the end, and that makes it an aldehyde. And so, and that's just the name of that molecule. But they both have a carbonyl group. So I always think of end, like that, an aldehyde, so they have similar beginning sounds, and so therefore it helps me to remember. It helped me to remember that. And so these guys can be structural isomers with different properties, ketones and aldehydes, and so that's the carbonyl group. Moving on, we have the carboxyl group. So the carboxyl group, notice here it has a C with a double bond to an O, but it also has an OH. So therefore, when looking at this, you have to really be careful and not look and say, oh, there's a C with a double bond, and oh, that must be a carbonyl group, because if it has the OH group, it's not the carbonyl group, it's the carboxyl group. All right, and so when we look at this here, it gives the molecule acidic properties. So they're called carboxylic acids or organic acids. The key word is that it's an acid. So this is what it looks like attached to a molecule. So this is acetic acid that's in vinegar. So it has acidic properties. What does it mean to be an acid? It releases what in water? It releases H pluses, so it's a source of hydrogen ions. And so it gives off hydrogen ions. So what happens is, as it says here, the covalent bond between oxygen and hydrogen is so polar that hydrogen ions tend to dissociate reversibly, meaning they can go back on. And so therefore this goes back and forth here. And so here's your acetic acid. What happens here, notice that, notice here, the H that was once here is now released, and it's released as a H plus, leaving the O with a negative, meaning that the hydrogen is one electron. It leaves the electron with the oxygen, so the oxygen's outer shell is still filled, but it has a slightly negative charge, and that hydrogen is now missing an electron, so that's H plus, and so it releases in this direction, releases H pluses, and so in this direction it acts as an acid, releasing it, all right? And so that's its function. And so that's an organic acid or carboxylic acid. The amino group is next. So the amino group looks like NH2, and so this is what it looks like attached to a molecule. So notice that this molecule is what we call an amino acid. So we've talked about this before. So it has an amine. It's an amine because it has the amino group, so that's the name here. Here's the amino group. And it's also, notice here, it has the carboxyl group that we just talked about from above. So it has the carboxyl group, and therefore it acts as an acid by releasing H pluses, by the same token that we just talked about up here. But the amino group makes it act as a base. So this right here is the amino group. And remember how bases work. They can either release OH negatives or take in H positives. And so it acts as a base and can pick up a proton from the surrounding solution. So here's your NH2 here attached to the rest of your molecule, but it actually can pick up the H's, all right, here. So if an H plus is floating around in the solution, it can pick that up and therefore have a slightly positive charge because of the H pluses here. And so that's what that is. And so therefore, by it accepts H plus ions from the solution it's in, therefore decreasing the hydrogen ion concentration in the solution. Decreasing the hydrogen ion concentration does what to the pH? It increases the pH or makes it less acidic, and so therefore acting as a base, so it's making it more basic. Making it more basic. Hence acts as a base up here. And so that's what an amino group here does. And so when we look at something like an amino acid, it has the two functional groups. It acts as a base as well as an acid, hence the name amino acid, having those two different functional groups attached to it. All right, and so then looking at this last page, the sulfhydryl. And so sulfhydryl comes from the element sulfur and hydrogen, so it has a sulfur and a hydrogen. Molecules that have this are called thiols, and so it looks like this attached to it. And these we'll see in more in-depth next chapter, but they can interact to stabilize protein structures. So amino acids can have sulfhydryl groups, and they can interact and make the protein very, very strong. And so right now I'm just going to introduce that to you, and helps to stabilize the protein structure. And then lastly, we have the phosphate group. The phosphate group has phosphorus in it, so that's your P here, and it has the 4 O's here, so it looks like this. And so when we look at this, they're called organic phosphates, and so this is what it looks like attached to it. And it makes the molecule of which it is a part an anion, because notice here it has these negative charges, negative, 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 negative. And so an anion is it means a negatively charged ion. And so it makes the molecules attached to an anion, and it also is involved in energy transfer. And you've probably recognized the term, or the word ATP, the molecule ATP. If you remember that, that's a major energy molecule, if you remember from biology. So ATP basically has a molecule called adenosine attached to 3 phosphate groups. So A is the adenosine molecule, triphosphate, TP, 3 different phosphate groups. And so the bonds between the phosphate groups is unstable because they both have a negative charge, and negatives, the same charges repel. And so therefore it's an unstable bond, and when the phosphate group breaks off, energy is released, and therefore this molecule is used for a lot of processes in cells that need energy. So one phosphate molecule called adenosine triphosphate, or ATP, is the primary energy transferring molecule in the cell. So ATP consists of an organic molecule called adenosine attached to the string of the 3 phosphate groups that come off of it, like my little picture here. And so we spend quite a bit of another chapter talking about ATP, but this just shows you like kind of an intro and makes this, that it can transfer energy between organic molecules, make a little bit more sense. And those, folks, are your functional groups, all right? And that is chapter 4 in a nice, neat little nutshell there, okay? Thank you very much.